Thank you for coming to my talk. Glucocorticoids correlate with and predict social status in the cooperatively breeding lance-tailed manakin, Chiroxiphia lanceolata. Cooperation is a specific type of complex social behavior. It's defined as an adaptation that has evolved, at least in part, to increase the reproductive success of the actor's social partners. The ultimate aspects of cooperation have been of particular interest to evolutionary biologists, because cooperation often involves seemingly maladaptive behaviors. In cooperative reproduction, for example, some individuals forego or delay their own reproduction to help others reproduce. Such reproductive roles are often determined by a strict social hierarchy. Efforts to understand the proximate mechanism that determines social hierarchies have revealed a hormonal correlate, glucocorticoids, a type of steroid hormone associated with the physiological stress response. They co-vary with social rank. Dominant individuals tend to have higher levels of glucocorticoids. A special thing to note here is that hormonal measures are thought to reflect a physiological trait that is more or less consistent across time and context, a physiological phenotype. However, does hormonal variation among individuals influence their position in the social hierarchy, or are these physiological measures merely a consequence of occupying a particular social class? Before we get into how we sought to address these questions, let me first take some time to talk more about glucocorticoids. As I said before, they're steroid hormones. They're well conserved among vertebrates. Corticosterone, which is more commonly referred to as cort, is the primary glucocorticoid in birds. Cort is an important hormone that circulates at baseline levels. However, stressful stimuli can increase its concentration via activation of something called the hypothalamic pituitary adrenal axis, which leads to an elevation of cort concentration well above baseline levels. And cort has a myriad of physiological and developmental functions, including the breakdown of stored energy and the modulation of the immune system, among others. Cort also correlates with behaviors and cognitive abilities as well, including aggression, exploratory behavior, spatial memory, anti-predator learning, and neophobia. We investigated the role that cort plays in social hierarchies in the lance-tailed manakin. Now, this bird is a small, frugivorous, non-migratory neotropical passerine. Pictured here are the different plumage stages of males. Males have a delayed plumage maturation. They don't reach their full maturity until their 4th year. Second and third year males are termed subadults, and females look very similar to the hatch-year male that you see on the left. And what you see in this video are two enthusiastic males that are displaying for a female. This is known as cooperative courtship, which is a particularly puzzling behavior. A dominant alpha and a subordinate beta form a partnership in which they cooperate to advertise a territory and attract females, with coordinated courtship displays. Yet alphas capitalize on the mating opportunities of over 99% of the time. As a result, cooperative courtship in lance-tailed manakins is an opportune model to explore the evolution of social hierarchies and the mechanisms by which sexual selection produces seemingly costly phenotypes within that hierarchy. Young males have a few options as they mature. They may become an alpha, the top of the social hierarchy right out of the gates, or perhaps they'll take a more indirect path, starting out as a beta. A third option is to do neither, wandering around as a marginal male with no territory or steady partner to dance with. Now, it's important to note here that an individual may fill each of these three distinct roles throughout their lifetime, usually moving up the social hierarchy. Now, this variation in social status in males is at the heart of what we're trying to figure out. What determines who becomes alpha and when, or who never moves on beyond marginal status, and so on. So this brings us to our specific questions. First, are individuals consistent in their cort profiles? That is, can we claim a hormonal phenotype? Second, does male cort vary with status? And third, can early life cort predict later status changes? Challenging the idea that high cort responsiveness is merely a reflection of holding a high social status. Now, lance-tailed manakins studied for this project are located on a 46-hectare study site on Isla, Boca, Brava. an island off the Pacific coast of the Cherokee province of Panama. This banded population has been monitored by my co-author, Emily Duval, for over 20 years. In any given year, this population has approximately 30 individuals. Now, the social status of males are determined by specific criteria based on anywhere between 800 and 1,000 hours of in-person behavioral observations that we do at focal territories every year. Alphas are defined as the most commonly present male at a territory that performs solo displays with unique display elements, such as the eek display that we have here. And this is a display that precedes copulations. Betas are defined as the most common duet song or dance partner. And marginal males are non-alpha and non-beta males. They may engage in some cooperative courtship behaviors with other males, but not regularly and not typically when females are present. Now, we measured court in the field using a standardized capture and restraint protocol, which is widely used. Lance-tailed manakins were captured using passive mist nets. An initial blood sample was obtained within 2 to 3 minutes of the individual hitting the net to ensure a baseline sample could be quantified. Subjects were held in a bag for 30 minutes, removing them at 15 and 30 minutes to obtain stress-induced samples to create a time series of court concentration. Now, we quantified the area under the curve as an individual's stress-induced measure, or as we call integrative court. Over the course of 3 years, we captured 173 unique individuals, males ranging from subadults to alphas as well as females, many of which we sample more than once. To determine if lance-tailed manakins exhibited a hormonal phenotype, we conducted a repeatability analysis that uses the intra-class correlation coefficient, denoted here as R, as a measure of repeatability. We found that our stress-induced measures of court were repeatable within individuals across time, with an R of 0.34, which is right in line with the average measure of repeatability of stress-induced court found in a couple of recent meta-analyses. However, the same cannot be said for baseline samples, as they are not more dramatically, as they're likely more dramatically influenced by acute changes as per the circadian rhythms and metabolic status. I've included a heat map here of the coefficient of variation for each individual who was sampled at least twice over this 3-year period. Now, the redder the line indicates the more widely the individual's court measures vary across time. So you can see that the variation is much lower for stress-induced measures on the left relative to the baseline measures on the right. Next, let's take a look at whether there was a correlation between baseline court and social status. Social status is on the x-axis, including females, and the y-axis is our measure of baseline court concentration. Groups that share like letters above the box plots indicate no statistically significant difference. So alphas, as you can see, have a higher baseline concentration on average, whereas the difference among other groups are not statistically significant. We have a similar setup for integrative court, which is our stress-induced measure. Alphas respond to a standardized stressor with higher concentrations of court compared to all of our other social groups and females. There's also a difference among our non-alpha males. Subadult males have on average the lowest concentrations of stress-induced measures compared to marginal and beta males. Now, similar to other studies, we found that stress-induced measures of court are repeatable, suggesting a court response phenotype, while baseline measures were not repeatable. And similar to studies in cooperatively breeding species, court positively correlates with social status. Because stress-induced court is repeatable, we went beyond a simple correlation and asked whether court responsiveness during early life was related to attaining status later in life. Based on the correlation, we predicted that subadults with low court responsiveness were more likely to become marginals in the first few years of their life, while high court responders were more likely to become alphas. Here we have the results of a logistic regression with integrative court of subadult males on the x-axis and whether they achieved beta or alpha status on the y-axis. Alphas are colored red and betas are blue. And as you can see, individuals with higher stress-induced court measures as subadults are more likely to obtain a beta or alpha status within the first few years of their lives. Or I should say the first few years of becoming fully mature. Now, this is based on 5 individual males that achieve status, and such is the nature of studying long-lived species in the wild. So while these results should be taken with a reasonable amount of caution, they do, however, make the case for further exploration. Indeed, our findings match the predictions that subadults with low court responsiveness were more likely to become marginals in the first few years of their life, while high court responders were more likely to become alphas or betas. In summation, we found that court responsiveness is repeatable and correlates with social status and may predict future social status. This predictive quality of future social status suggests that high court responsiveness is not merely a consequence of attaining a high social status, but rather high court response phenotypes might have a higher predisposition for achieving high social status. We'll continue to monitor this population and track the individuals that we have physiological data on to see how it may or may not continue to correlate with their life history trajectories. We're also interested in how court might link to other physiological measures that influence longevity and performance, such as telomere dynamics and oxidative stress. Also, can the court phenotype be manipulated to induce a specific social trajectory? Perhaps this is feasible using manipulations during early development in ovo or in the nest. These data and results could not have been possible without the hard work of many graduate students and field technicians in the Duval lab and grants awarded to Emily Duval that funded the research. Thank you very much for coming to my talk, and I'm happy to answer any questions.